Hey Salma, today I'm going to be going over what we didn't get to go over in our session earlier today. Going to be going over Mendelian Genetics Concept 2, starting off on slide 12. So, we're going to be starting this off with the law of independent assortment. In a dihybrid cross, each pair of alleles segregates independently of any other pair during a gamete formation. So, that's basically saying that your two chromosomes become one. Um, This law applies to genes on chromosomes that are not homologous or those that are far apart on the same chromosome. So, if you'll look in this little diagram here that, let me zoom in because I can't read that subtext. Uh, it's easy to see how genes A and B can be shuffled into different gametes because they're very far apart and they're on different genes. Um, Whereas genes C and D, they're really close. It's going to be hard to like shuffle them and independently assort them. They're called linked genes, which means that usually inherited together. Um, so independent assortment alignments. So independent assortment of genes in different chromosomes reflects the fact that non-homologous chromosomes can orient in two ways that are equally likely. So, oh, okay. So in this example, the resulting gametes can either be both two that are big A, big B, and two that are little b, or you get two that are big A, little b, and two that are little a, little b, or little a, big B. So, how many unique gametes can be produced through independent assortment by an individual with the genotype big A, big B, big C, little c? So you can do what Dr. Briggs likes to call the brute force method, which is just going through each possible combination, or you can do the shortcut using math. So if you notice that the A gene can only produce one possible thing, a big A. The B gene can do big B or little b, and then the C gene can do big C or little c. So 1 times 2 equals 4 possibilities. So um, n equals the number of loci, or n equals the number of heterozygous loci, then the number of possible gametes equals 2 to the n power. That's a good one to write down. So let's go back to blood types. I know we went over that this afternoon. So blood cells have another kind of protein on their surface called the Rh factor. Um, Possible genotypes to determine that are big D, big D, or little d. Um, So in this example, parent one has blood type B positive. So it's big I B and then a little I and then big D little d. Parent two is blood type AB positive, which is big I big B, and then big D little d. So if you'll remember with our dihybrid crosses, you can figure out the possible genotypes of the gametes of these parents and the possible genotypes of the offspring of these parents. So, um, this here is just a little chart on blood groups. I think we went over that earlier this afternoon, and a chart of by race of what the blood type percentage for each race are. Yours truly would fall under 33% of the Caucasian population. I am type A positive. Alrighty. Rogam, Winrow, and Winrow. Um, I'm pretty sure that this is just a way you can like tell what your child's blood type is going to be before they're born. Um, oh, no, okay, so the mother and the child have two different blood types, the baby, um, it doesn't usually affect the mother as much, but she produces antibodies that will go after the baby's red blood cells because the body thinks that there's some sort of intruder in the system, and that can put the baby at risk for something known as HDFN, hemolytic disease of the fetus and newborn. Rogam prevents the mom from making antibodies by um, basically putting like getting the body accustomed to that blood type in a way. It prevents her from making antibodies. So, um, yeah, good stuff. So solving complex genetic problems with the rules of probability. Um, so we can apply the rules of probability to predict the outcome of crosses involving multiple characters. So a dihybrid or other multi-character cross is equivalent to two or more independent monohybrid crosses occurring simultaneously. So when you calculate the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. So say I have a 1 in 2 chance of flipping a head on a coin, and then I have a 1 in 2 chance of flipping a head on a second coin. I then multiply 1/2 by 1/2 to get 1/4. I have a 1/4 chance of getting a head on both coins. Um, and then the example given here is um, if we cross, I'm just going to scroll all the way down. All right, so if we cross F1 heterozygotes of type big Y, big R, you can calculate the probability of different genotypes among the F2 generation. So, what's the probability of a fully true breeding offspring? Big Y, big R, big R. Fully homozygous dominant. So the probability of big Y is 1/4, and the probability of big R is 1/4. So the probability is equal to 1/4 times 1/4, which is 1/16. So what's the probability of a fully heterozygous offspring? Well, big Y has a 1/2 chance, and big R has a 1/2 chance. So 1/2 times 1/2 is 1/4. There's a 25% chance that they're heterozygous. So for example, for the cross big P, big R, and big P, little r, you can calculate the probability of the offspring showing at least two recessive traits. Or so the probability that the offspring shows at least two recessive traits. So it's either going to be little p, little y, little r, or little p, little r. Uh, so, um, the chance of it being little y, uh, and then big R, little r is going to be 1/4, which is the probability of little p, 1/2, which is the chance of little y, times 1/2, which is the chance of big R, little r. And that gets you 1/16. So you just go down the line, and it'll get you 6/16 or 3/8 of a chance. So, skip through this. So, Extensions to Mendel. So codominance, we went over that. That's with blood types. And incomplete dominance, which is with the flowers. Genetic polymorphism. So genes often have more than two alleles. Um, that's like in blood types because there's three genes. There's big I, big A, big I, big B, and little I. Each individual will still only get two alleles. There's just now three alleles to pick from, um, rather than two. Genes with multiple alleles. So, um, rats on slide 35. Um, they have a gene that codes for their fur color, called agouti. Or agouti is a type of their fur color. So, um, there's a lot of different rat fur colors, and there's a complex dominant. Yellow is the most dominant, followed by light agouti, then agouti, then black and tan, and then black. Um, that the A to the Y is yellow, A to the 1 is agouti light, to the plus is agouti, a to the 1 is black and tan, and then little a is black. So, pleiotropy is one allele has more than one effect on a phenotype, and so that's with the mice. So in this example, Q-Not's yellow mice. He tried and tried and tried, and he could never get a true breeding, fully homozygous line. Um, the big Y allele is dominant over the little y for color. So little y is a white mouse. Big Y, little y is a yellow mouse. However, the big Y allele is recessive to the little y allele for lethality. So little y, and big Y, little y, the Y allele is dominant for development, but big Y um, means that both alleles combine to form a lethal combination. So that's why he never found any like fully true breeding mice, is because the ones that were big Y either died in the womb, or died during birth, or died like shortly after birth. Like they were never gonna make it. And that's what pleiotropy is. In this example, it controls fur color and if you're gonna live or not. So epistasis is one gene interacts with the expression of another gene. So in this example, dogs. Everyone likes dogs. Um, specifically Labrador Retriever coat color. So the products of some genes interact with each other and influence the phenotype of an individual. So the E gene controls whether or not there's a coat color or not. So big E, and big E, little e code for dark color, and then little e just codes for a light coat color, and that's what results in yellow labs. So if there is a dark color, the B gene controls for how dark. So a, the dominant E gene, or the dominant B gene, makes a black lab, and then the recessive B gene codes for a chocolate lab. Um, so in this little example, on slide 38 shows the Punnett square. Rather than your typical 9 to 3 to 3 to 1, um, that happens when you breed two heterozygous individuals. This example shows a 9 to 3 to 4. And that's what's going to be seen when there is epistasis at hand, is that the presence or absence of one gene controls, um, interacts with the expression of another gene. So if that E gene is recessive, then the B gene doesn't matter. It's always going to be a yellow lab. So, although alleles assort independently, some genes can have an influence on the expression of other genes. That's epistasis. So the white leghorn is white because the inhibitor allele F blocks the expression of the pigment allele C. Um, Then the white Wyandotte is white because the pigment allele C does not produce pigment. They breed, and most of their offspring are white, but they have a couple brown ones. So genotypes of the form big C and then that's I, not F, my bad. But the form big C and then little I, they have colored feathers. Um, so the result is 13-3, which is very different of 9-3 to 3-1. The chickens are brown because they have the combination of they don't have the color inhibitor, but they do have the pigment gene. So, um, number 6 on slide 40, polygenic inheritance. Um, Multiple genes control the phenotype of a trait. So, these traits show continuous variation and they're referred to as quantitative traits. So, the example is human height. Your height is determined by a lot of genetic factors, and environmental. But yeah, polygenetic inheritance, multiple genes control one trait, whereas epistasis is, or not one, epistasis, pleiotrophy is one gene controls many traits, or multiple traits. Could just be two, it's just more than one. Um, and then number 7, gene expression is influenced by the environment. So, coat color in Himalayan rabbits and Siamese cats. So, an allele produces an enzyme, and that enzyme allows pigment production only at temperatures below 33 Celsius. So, it'll produce that pigment when it dips below a certain temperature so that the animal can get warmer faster. Otherwise, it's just going to stay white. All right, tracking traits. Um, Pedigree analyses. Some human traits are controlled by a single gene. Some of these exhibit dominant inheritance, and some exhibit recessive inheritance. Pedigree analysis is used to track the inheritance patterns in families. Um, Humans are not good subjects for genetic research. We have way too long of a generation time. It can take anywhere from 20 to 40 years for an organism to reproduce. Um, Parents produce relatively few offspring, especially these days. You don't see too many families with more than 2 or 3, sometimes 4 kids. Like, it's a wonder that, like, you see families with 5, 6, not gonna say it, kids. Um, and breeding experiments are unethical. Um, I'm pretty sure Dr. Briggs already went over ethics with you, or you've done plenty of science classes involving ethics. Um, it's bad. It's morally wrong to experiment on kids. That's why, um, yeah, don't experiment on people unless it's for psychology reasons. Then it's okay. Don't biologically experiment on people unless it's for a legal, good drug trial. Don't biologically experiment on people without them knowing and negatively affecting their well-being. Um, so in pedigrees, a horizontal line represents individuals mating, a vertical line leads to the children. The children are arranged horizontally, left to right in birth order, and then a double line means mating between relatives. Um, a lot of the royalty used to do that. It's not good. So, the pedigree of a dominant allele. This shows the patterns of brachydactyly, which is, in this example, your thumb and your middle and ring fingers are normal length, but then your pinky and your index are like really, really short. So, for a rare dominant trait, most matings that produce affected offspring have only one affected parent. For matings in which one parent is affected, about half of the children are going to be affected. Um, in a one of these. trees. If the circle or square is shaded, that means they have that trait. Um, Affected individuals appear in each generation, and affected individuals are equally likely. So if they appear in each generation, that's pretty solid evidence that this trait is dominant rather than recessive. And if it shows equally in males and females, then it's not a sex-linked trait. You're going to get into that in genetics. So the pedigree of a recessive allele. So a rare recessive trait skips one or more generations, um, and affected individuals um, that often result from the mating between relatives. And affected individuals can have parents who aren't affected. All right, so that's going to be it. Long video today, Jesus. Hope you enjoy. Bye.